Okay, so welcome everybody. My name is Florian Gehre. I'm from the Bernhard Nocht Institute for Tropical Medicine in Hamburg, and welcome to our symposium here about mobile labs. Also welcome to the speakers, then Mona, Laurence, and Kim, and myself. So I think without giving a long speech, we just start. And I prepared a few slides, just the introduction about mobile labs. Then I will present about a mobile lab project that we have in Europe. And then we have two presentations about mobile labs in Africa. And then we have also mobile lab activities in Canada, so to cover a little bit a geographical spread. Let's see if this will work. Okay, it doesn't, huh? How does that pointer? Did it work? Okay. Yeah, so basically we started. So what are mobile labs? So mobile labs, is the name basically already kind of gives it away, are mobile laboratories that you can send into remote areas where we can provide diagnostic services for outbreaks, for surveillance, or also for research if you want to. And here the key is really that outbreaks normally happen in very remote places, and in Africa at least, or also to a certain extent in Europe, you have then to refer these samples into central reference labs, and it can really take like a long time, like up to days, for a result to come. So the main purpose of a mobile lab is really that you can reduce this diagnostic turnaround time and provide same-day patient results, and then also give obviously like a kind of epidemiological overview for public health practitioners and interventions. So on the right side, the figure shifted a little bit, but what you can basically see here, we looked at some data over the years, since last 15 years or something, the number of mobile labs really increased in Africa and in Europe, so they're getting more and more important. And I think acknowledging this, also the WHO a few years ago started to develop some guidance documents around mobile labs. They call it Rapid Response Mobile Laboratories, RRMLs. And within this guidance, they started with a classification system, because these mobile labs can come in all kind of sizes and shapes. You can see we have 5 types. On the left side, this type 1 labs. These are really suitcase-based labs where you have like 1 or 2 boxes maybe. Type 2 labs are getting more complex already. These are like 15, 20 boxes. You can carry them around, ship them around, and set them up in tents. And then you have like labs in small trucks, medium trucks, or even labs in fleets of trucks. And ultimately, as you can imagine, with increasing type number, basically, the complexity of operations are also getting higher. You can do bacterial culture in these truck container labs. And I think the ultimate goal of this exercise then is to develop some global guidelines or standards, which is at the moment ongoing. And these standards include like all kinds of aspects of mobile lab operations, and should guide people that want to maybe develop a new lab, or also countries receiving a lab, to make sure that this is not an accreditation but more a recognition system. So make sure that these labs fulfill certain standards, that you can be sure whatever is operating in your country is okay. So yeah, as I said, today basically this session is about giving you a little bit of an overview what labs are out there. As I said, we have labs from Europe, an initiatives from Africa, basically from hot regions, conflict areas, and then also labs from Canada, where they operate mobile labs around the Arctic. So in very different conditions. So we want to show you a little bit how to set them up maybe, and then also how to integrate them potentially in some kind of national systems. Yeah, so let me start with my presentation then about European lab. We have a project funded by the European Union that is called MOBILISE, and here we focus on a novel lab that is green and especially made for One Health purposes. That lab is meant then to really act on the fact that, as we all know, that with changing weather patterns, we know that spread of vectors like mosquitoes and ticks is increasing all over Europe. And with these vectors, then also we have the spread of novel diseases into Europe, arboviruses, and this is where our lab is getting important. Within this project, we want to focus on two pathogens, basically West Nile virus and Crimean Congo hemorrhagic fever virus, risk group 4 and risk group 3 pathogens. And as you can see here, you have basically vectors and animals involved in the life cycle of these pathogens. So these are zoonotic diseases, and this is where this One Health aspect would come in from a laboratory perspective as well. In Europe, as well as in Africa and many other regions, but there are still some key challenges for these zoonotic diseases. Obviously, as I just mentioned, the One Health aspect needs to be considered, or should ideally be considered. We have often in countries we don't have BSL-3 or BSL-4 labs that could diagnose these pathogens. And since these are somehow climate change-induced pathogens in the end, or emerging pathogens, it would be nice if your lab would be operating in a green way. And these are the three gaps that we try to then solve, basically. We have a consortium of 9 partners across Europe. We, as the BNITM, we are the coordinators. We work together with the Austrian Institute for Technology in Vienna. We have Exos from Greece, which is a software developer. We have Bayer Consulting from Romania, and MDSC in Estonia. They're actually building this lab. And then one key thing from the call was that we need to work with national agencies. So in that sense, we have the Friedrich Loeffler Institute from Germany, the Austrian Agency for Health and Food Safety, AGES. We have the National Public Health Organization in Greece, and then also the Red Cross in Romania as partners. So let me show you some pictures of the lab that we then developed together. As you can see, it's a lab that is mounted on a truck. It has like a 20-foot container footprint, basically. It's not really a container, but it has this size. This can expand it on both sides basically. And ultimately, with a 20-foot container footprint, we have almost the size of 3 20-foot container workspace. And as I said before, we wanted to make this green, and you can see this thing is basically covered in solar panels. Ultimately, we have 4.5 kilowatts of solar panels. We have 2 times 200 amp hours of battery backup that can obviously then also be charged through solar. You can't see this, but there's also the possibility of setting up a small wind generator. If that one makes much energy, I'm not sure, but we will try that. And then it can also obviously be connected to mains power, and then diesel generator for emergencies if really all powers would fail. We are establishing smart workflows. So by that, we try to really manage peak consumption of the lab equipment so that you don't run, let's say, the autoclave and all the other equipment at the same time, so that you make sure that maybe the solar power can cover everything, and we really put a focus on buying and installing energy efficient equipment. That means also heat pump system, but also really energy efficient lab equipment. If you have a look inside, basically we have 5 working areas. On the left side here, this area 1, what we call, as I said, there's a One Health mandate to it. So we have a BSL-3 tent that is basically for sample reception outside. So here you could dissect animals. You can receive human samples anyways, but also dissect animals, birds, and subsample them. And then once you have these subsamples, you bring these into the main lab. You can see in the center here then the main lab with the wings on both sides. And in the middle is a blue room. That is a BSL-3 room with a negative pressure and an airlock. And inside that room on the bottom left, you can then see that there is a glove box for inactivation of samples, as well as a biosafety cabinet, an autoclave inside, and then incubators as well. All water from the airlock, all hand wash water can be decontaminated with an effluent decontamination system. And then in the wings, basically, we can set up, or will set up in our PCR and our sequencing workstations. And the nice thing is they're physically separated, so we could really do a pre-PCR, post-PCR, or pre-amplicon, post-amplicon setting to avoid cross-contamination. And so this is actually quite nice, I think, about the lab. We also have a VITEC in the lab so that we can do automated microbiology. Yeah, so again, this is the overview of the diagnostic portfolio. We have PCR machines. We use then, or want to validate lyophilized kits. So again, this comes back to this energy efficiency. The lyophilized PCR kits, they don't need any fridge or freezer, so they can be stored like basically in a drawer, which will be, if it works, this will be very nice. And then we have a sequencing platform, an ISAC, and then, as I said, microbiology with the VITEC. Our partners in Austria from the AIT, they are developing also new rapid tests. At the moment, the targets are West Nile and Crimean Congo, as I said. These rapid tests ideally are then DNA or RNA based, so they're detecting directly the virus. And they are automatically readable through near-field communication and or other methods like just scanning with the phone. But the nice thing is you could then do this directly in the glove box. Yeah, you get the result out of the glove box without having to export anything. So this would be a huge acceleration of our diagnostics. So ultimately, our lab should then be included in the national sampling strategy from the national agencies. They can bring us human, veterinary, or environmental samples. We can inactivate them or culture bacteria, and we can sequence and PCR them. So one thing is obviously the hardware, but we are now also doing is that in February 2025, so in 3 months, we're going to have a mobile lab training then in Hamburg to develop really a pool of operators. Here we invite then people from our national agencies, ARGES, FLE, and the MPHO, as well as colleagues from the National Public Health Lab in Tanzania. So they get trained then in operating this mobile lab, and they are meant to be the multipliers to train more people in their countries. And also for bioinformaticians that are trained then in the analysis pipelines that we want to implement or implemented now. And then in the next year, so the project runs 1 more year, we then have field missions to really field test this lab and field test everything around it, as well as also the trainees. And in Austria, we will test the Crimean Congo workflows. In Greece, we will help them in West Nile outbreaks because they are now annually. So this will then obviously be operational in Greece, really. And then in East Africa and Tanzania, we are going to join the National Public Health Lab in Rift Valley fever surveillance activities. And we have still a 4th mission that we are currently planning. So ultimately, we hope that we have something like 10 to 15 European operators and 4 African operators in this pool of mobilized people. Yeah, and then let me conclude. Basically, I think in this project so far, we were able to develop this One Health BSL-3 lab, as I said, a small footprint versus a kind of large working area, and we can do then these pre and post things. And we hope it will be green, which I'm very confident that the solar will actually power most of our equipment. Yeah, I think that's from my side for now. These are our partners that I want to acknowledge. Yeah, thank you. So I think we could take questions now. If anyone has questions, maybe we can take 1 or 2, and then I would hand over to the next presenter. If there are no questions, it's also okay. But okay, I see a question there. I think there are microphones.
Okay, thank you. I come from France, and I'm really interested by this possibility of mobile laboratories because, as you know, France has overseas territories, and that introduces a lot of complexity when dealing with the microbiology issues of outbreaks. I was just wondering about a very practical question. What is the budget necessary for maintaining one of these mobile laboratories annually and for deploying it? Just a curiosity so I can bring the answers back home and discuss it.
It is a very complex question.
Yeah.
We can discuss this, but I think for maintenance, we have other projects. I mean, Muna is going to present it in a second. And for example, the other projects are included in the operations of the governments, of the ministries of health, and then the maintenance is mostly covered by them. And I think the way we choose the equipment, it doesn't normally need much maintenance and should be quite robust in the mobile labs. Most expensive are actually the per diems you pay for your laboratory operators in, let's say, in Africa, which I think in Europe is not so important. So this is from our experience the biggest expense normally, and then operating it is similar to a normal lab. Yeah. There's one more question.
Thank you. I'm Arne from Norway, Norwegian Institute of Public Health. So a very interesting talk and a very interesting concept. I was also interested in more like practicalities. Could you say a little bit more about building this lab, this truck, and how many have you built? Is it one prototype now, or?
Yeah,
and I imagine there are a lot of challenges doing this.
Yeah, this is now a prototype that we built. We are basically the lab experts, and we work together with this company in Estonia that are part of our consortium. They are the engineers. They normally don't build labs, so this was challenges here to find a common language between biologists or whatever and then engineers. I think overall, yeah, it's a prototype. We would probably, we started putting this, for example, already on a smaller truck before, but then it became too heavy. So the Mercedes truck you see there is now already the first improvement. And I think once we go to the field, there might be still improvements around the whole thing, especially when it came to the weight distribution, I think, but the bigger truck takes care of this now. But I think we will find out challenges still in the field. Also around the solar, we have to try it. If we operate it in Greece, then we need air condition in the summer, and then yeah. Okay, so maybe I'll hand over then to the next presenter. Welcome, Mona Affara. You can introduce yourself. And
you. So my name is Muna. I'm also from the Bernard Noch Institute for Tropical Medicine, but I'm going to give a different perspective and present on a mobile lab network we've established in East Africa over the past 7 years. So this is a bubble plot from the WHO, and what we've seen in Africa is an increasing frequency and magnitude of disease epidemics of international concern. And we see this trend in the East African countries that we're working in. And as Florian's mentioned, one of the biggest challenges is that often these outbreaks occur in the periphery, where there's limited or no diagnostic capacity, and samples therefore have to either transported to the central laboratories, or in some cases, they need to go to neighboring countries for diagnosis, and this takes time whilst the outbreak continues, transmission continues. So the mobile lab offers a solution to this, bringing the diagnostics to the outbreak and therefore supporting containment efforts. And because of this, the German ministry, through the German Development Bank, KfW, decided to support the establishment of a mobile laboratory network in East Africa, implemented by the East African Community. So this is an intergovernmental body comprising of 8 partner states, which are Tanzania, Kenya, Uganda, Rwanda, Burundi, South Sudan, and more recently, the DRC and Somalia have joined this regional economic bloc. And at BNITM, we have partnered with the EAC in the implementation of this mobile laboratory network over the past years. And in order to achieve this, we're working with the national public health laboratories in each of the EAC partner states. So they have the mandate for outbreak response in their countries, and they are the recipients of our mobile laboratories as well. So the project has been operating and can be divided into 3 phases. So in the first phase, we've really focused on modular box-based mobile laboratories mainly for viral hemorrhagic fever diagnostics. In the second phase, we're establishing container laboratories. So these are BSL-3 containers so that we're able to conduct bacterial diagnostics really for AMR surveillance in the region. And in the third phase, we are actually integrating the veterinary sector into our mobile laboratory network so we can have a more One Health approach. And for the implementation, really, there's 3 main areas we're focusing on. There's the project governance, which is important for it, the diagnostics portfolio, and the capacity building. So when we look at the project governance, this is the structure for our project. So the mobile lab network is integrated into the Ministry of Health. As mentioned, the National Public Health Labs here, they're the recipients of our labs. And together with the EAC Secretariat and BNITM, we form the Regional Technical Expert Working Group and a steering committee which meets twice every year, basically, to drive the direction of the project and make decisions. And the information from this is fed into the sectoral councils of ministers of health and then up to the Council of Ministers for decision making and eventually to the heads of state summit. So decisions are made at a high level, which really helps with implementation and sustainability of the project. And when we look at the diagnostic portfolio, as mentioned in the first phase, this has really been focused on viral hemorrhagic fevers. So in the modular labs, samples, infectious material is imported into the mobile glove box where it's inactivated, and then the downstream diagnostics is RT-PCR and serology. In the second phase, we've integrated automated systems for blood culture and bacterial identification and susceptibility testing. And in the third phase, we're integrating all of these diagnostic capacities into the veterinary sector. And we've also established a next generation sequencing platform. And so this is basically an overview. So the platform we've established now is based on the Illumina sequencing platform within the region. So we've installed higher throughput NextSec 1000 sequencers into the National Public Health Lab. So these are the stationary sequencing capacity. And then in the mobile labs, we have the smaller, lower throughput ISEC 100 units. So therefore, we have capacity both for pathogen identification in an outbreak, as well as for AMR surveillance. And then we can do also genomic epidemiology within the East African region. And parallel to all of this infrastructure has been quite a comprehensive training program to build capacity in the region. This is important. So the training is both for the lab experts within the National Public Health Laboratories, as well as for other operational aspects of the mobile laboratory. So, for example, driver training and training for biomedical engineers within the National Public Health Lab so that they can maintain the equipment. And this is basically an overview of the trainings that have been done. So we've conducted quite a number of trainings, wet lab, bioinformatics, sample shipment, to cover all aspects. And now we really have a pool of over 400 experts in the region that are trained to be able to operate the labs in the different EAC partner states. And we handed over the phase 1 labs to the partner states in 2020 during the COVID pandemic, and they were immediately deployed, mainly to the border regions in each of the countries to help in screening individuals crossing the borders. And then since then, they've really operated in over 50 different field missions within the region. These have been preparedness missions, also training, surveillance, and outbreak response. So, for example, the labs were active in Uganda during the Sudan virus outbreak, the Marburg outbreak in Tanzania, and currently quite a number of the labs are active with mpox within the region. So just in summary, some of the lessons that we've learned so far from the implementation of this project: sustainability is important. And in order to achieve that, I think it's important to integrate into the national system so that the labs are considered both in the task force for outbreak response, but also for funding as well, so that they're part of the annual budgets within the country. It's also important to have a holistic training approach, so not just focusing on the lab experts, but also all aspects of the mobile lab. For a rapid response, we realize supply chain management is important, so the procurement of kits. So we've established a system in the EAC, centralized procurement, and then good communication with the partner states to ensure that we clear goods very quickly, and this works for a rapid outbreak response. And also that the labs need to really be flexible. We use them both for preparedness, they're active a lot, preparedness and response. So for example, Tanzania deployed their mobile lab in response to the Ebola outbreak in Uganda, and as a result, they were able to rapidly identify Marburg whilst they were in that preparedness mode, and therefore move to a response. So I think this is very important as well. And it's important that the labs are accepted, and therefore we need good quality systems within the labs. So integrating them into existing ISO accreditation programs within the national public health labs, and having EQA programs within them, and also, as Florian mentioned, the minimum standards that the WHO are establishing that we comply. So ultimately, we have a network now of mobile labs that can rapidly provide a first-line response to outbreaks in the region. Thank you. See? I don't know if there's questions.
Hi. Honest question, I'm not trying to sell anything, but why Illumina and not nanopore, for example? Seems much easier to transport a mini-ion and the kit for the rapid barcoding than all the cartridges and the...
We were deciding between the platforms, but actually it comes down to, I think, the longer term sustainability, and basically nanopore doesn't really have a footprint. Certainly not within the East African region, for basically the longer term sustainability, whereas Illumina does have that. So they're able to support the national public health labs, both for maintenance, training, etc. So that was one aspect. And then also in terms of just for reliability as well, I think we decided that the Illumina platform would be a safer option.
Okay, thank you.
Hello, Ettore, ECDC. What, if I understand well, Rwanda is certainly part of the Eastern African community. The recent Marburg virus disease outbreak was pretty large, and the country experienced some challenges in testing, and it would be very helpful. Were you activated, and if not, what was the barrier from the government not to activate you? And then last question, when you need to move from one area to another, how complex from a logistic point of view it is? Thank you.
Yeah. So the lab was activated. Actually, in Rwanda, they actually have set the lab, some of the equipment in other key parts of the country. So in order to operate it, but so we've handed the lab over to the partner states. So they activate the lab under their outbreak response mechanism, and then we support them now as the EAC from both financially, but also technically to support their outbreak response. So yeah, it was activated in that. So we're not moving. The labs are not. You mean across borders, or I mean the labs were these labs were moved. Yeah, so the labs are modular, the box-based, the phase 1 labs, but we provided 2 Land Cruisers, so they have vehicles. One of them is like the ambulance one that can fit all the boxes, and then another one is a passenger one that you can then take the team in. So they have vehicles, but they can also transport if it's long distances and they want to fly. For example, in South Sudan, driving is very difficult, and they have deployed the lab. For example, in the yellow fever outbreak, they can also use flights. Yeah. I don't know you.
Hi, Laura. You've spoken a lot about integration with the national public health system, and I think now you briefly touched on nationalization. Do you have exit strategies with the way that you set up your labs, and do you hand them over eventually to the local Ministry of Health, or?
They actually already are handed over to them. So we are supporting technically, and we still provide trainings. But actually they are running their labs. And for example, often we might bridge financially for the first acute period of an outbreak, but then generally, actually, the national systems will sustain the deployment of the mobile lab. So in some countries, this is working more effectively than in others, but ultimately they are it's integrated into the national system.
And do you see sort of challenges with maintenance of quality control when you hand over after a certain period of time?
So actually, I mean, the national public health labs have they do regular EQA programs, and we are trying to make sure that the labs are mobile labs are integrated into that, and we establish, for example, for the container labs that are coming, we've already been doing a training and establishing local production of bacterial panels so that we can do like EQA for bacteriology within the region, because it's very difficult to ship these panels from internationally. So we're trying to develop systems for longer term sustainability. But it's obviously a concern, but this is
Let's just move to the next presentation. Otherwise, I see a countdown here. We are running out of time. So I just welcome then Laurence from MSF, and you're going to give us an introduction about your labs in conflict areas and also in Africa. So.
Okay. Yes. So I'm going to present, I'm Laurence, I'm the diagnostic advisor working in MSF medical department in Barcelona. So I'm going to present one experience we have, and not only the experience, but why did we run a mobile laboratory. So I'm just starting on putting the context. And I think it was already mentioned by our colleagues here, confirmation of an outbreak or detection of an outbreak is always a nightmare in MSF and outside of MSF. As you can see, the three examples, either you are sending the sample for the wrong pathogen, and because it's very vertical, if you send for measles, they will test for measles, nothing else. Or, and this is the case, was the case in Niger and in Ethiopia, that everybody thought it was a yellow fever outbreak. So you take the sample, you take time to go and see the patient, you take the sample, you send to the reference laboratory, it's already a challenge. The results are negative, so you need to go back and take another set to test for another disease, and it takes time. So basically, whatever Niger or the dengue outbreak in Ethiopia, generally it takes an average of 1 or 2 months to get the final result. If you are lucky, in many, because in many settings, we don't even receive back the result for several points. The most interesting one was the rubella one, actually, because everybody was convinced of a measles outbreak in DRC. So we took the sample, everything turned out to be negative. We thought it was because we are operating mostly in Eastern Congo and the reference laboratory is in Western Congo, and it can take 5 days only to transport the samples. So we decided to run again and to take those samples with particular precaution on how you take the sample. It turned out to be negative, a second one, but the operation in MSF say it's so evident it's measles. Let's move with the response, with the massive vaccination campaign until we receive a message from the laboratory. They test beyond measles and they test for rubella. At the end, it was a rubella outbreak. So you see also the impact of maybe the kind of operation and response to the outbreak if you don't have the correct diagnostic at first. So all these, definitely the rapid turnaround time of diagnostic are really critical for outbreak investigation. Where we are working MSF, but as it was clearly said by Muna, in many remote settings, you don't have the type of diagnostic you will need to confirm an outbreak. There are many deficiencies as well at national reference laboratory. I'm not saying that they are not able to do it, but sometimes they don't have human resources, sometimes they don't have the tools, sometimes they don't. For example, in Central Africa, they didn't want to test for hepatitis E because it's not part of the list of diseases they have to follow. So you see many complexity. But all these, and this is the most important for us and for many of us in this room, it's that by delaying the identification of the causative action of the outbreak, you automatically delay the response. And sometimes you can even put or response to the outbreak being totally out of the scope and the really target of the disease. So the question we had, and this is a bottleneck for the past 20 years, the question we had in MSF was then, would it be possible to have a laboratory that can go with an investigation, outbreak investigation team, and be able to run the test on site instead of sending the sample? And the second, I mean, from this question is basically, can we use the machine and can we demonstrate we can use machines that actually are in reference laboratory outside of this reference laboratory? So it's not so much to create a mobile laboratory, but to make people thinking that they might have already in this laboratory all the tools. It's just a matter of how to pack them, what are the logistic conditions you need to put behind in order to be able to bring this laboratory capacity jointly with your investigation team. So this is what we worked on. And let me bring you to, so it's in Central African Republic, Bangui is there. This is where you have the reference laboratory. MSF is all over the country, remote setting, conflict area, ongoing recurrent conflict for the past 40 years. Accessibility, so if you are taking, here it's the alert they saw the increased number of cases presenting in health facilities with fever and rash. So everybody say this should be measles. Okay, fine. If you take the sample here, can I transport by road, you can see some pictures which are not the worst. Congo can be worse as well. But the road, even if you don't have a conflict, it takes basically 10 hours for 60 kilometers. So you can imagine you have to drive for about 500 kilometers to reach Bangui, how long it can be. And then the only solution you have, it's NGO or United Nations plane, which is fine, but for the past 5 years, there is recurrent shortage on Jetta 1 for the plane. So we had to reduce the number of planes. So even if you take the sample, even if you have the place, you are not always sure you can reach the reference laboratory in the adequate time. So for all this, we say, okay, we need to bring definitely in this setup, we need to bring the laboratory. So what we did and how we did to develop, we start first thinking of what do we want? So we are focusing on alert investigation, which might be different from other type of laboratory on which kind of platform we are using. So we decide that it's going to be a mobile laboratory type 2, as it was shown by WHO registration, no, rapid response mobile laboratory classification. The technique we choose is ELISA. Why serology and not molecular? Because if you take, by the time you receive the alert from the field to your capital, you discuss with the people that it might be good to send an investigation team. You mobilize the investigation team and you go to the site. In many, many of the cases, the type of patient you're going to have are people with more than 2-3 weeks of symptoms. So we need also to choose the technologies that fit the target people. And it might not be the same in response, which is patient mobile laboratory for patient care, than for outbreak investigation. So it's look like this, no more. Those blue are the cold chain. We're using the indigo cold chain, which is a sustainable way to keep sample and reagent for about 7 days without recharging them. The problem is you need to have 5 units to transport 3 kits and store 20 samples because the inside is very small, but this is one solution. We wanted them, and the most important, we wanted self-sufficiency in terms of electricity and also cold chain. We manage for the cold chain. For electricity, we manage now to have and be able to run the laboratory based on the big power bank using solar panel, which decrease the use of petrol and so on. The problem is that transporting lithium batteries in plane is a challenge. So I think it's, so these are about 400, 500 kilos in total. It fits in a Land Cruiser. It includes also the structure, so it can be run in a structure, but also if you are in the middle of nowhere, you can have a tent to be able to run it. And after, it's the normal organization of the laboratory. This is how we test it. So we took about some months to design a bit the flow, especially working on logistics, because I'm not going to hide that in this kind of context, the outside temperature is about 35 degrees. So it might be difficult sometimes. So we work on the structure, how to organize. We had some improvements, especially focusing on reducing the temperature. We work another step was, now it was ready after testing in Bangui, so we simulate an investigation of outbreak in Power, our colleague from MSF France, basically to test the transportability. Are the boxes we are using strong enough to transport very sensitive material? And then the main result of this, it's after all those pilots, we had what we call the minimum viable product ready. And then since June 2021 up to June 2024, we had several real-time deployments. jointly always, and Muna was mentioning about the integration. This is also something that for us was extremely important, working this project and with the Ministry of Health and also with Pasteur Institute in Bangui. So the investigation team were mainly people from the Ministry of Health, and then only the laboratory team was somebody from Pasteur and somebody from MSF. And the good thing is we measured during all the development. What we wanted is to make sure that the results we got are in concordance with what we would have had in the reference laboratory. So we were systematically sending the sample in the reference laboratory, and in all deployment we have always offer over 95% concordant results. So I think the lesson learned is first we demonstrate clearly the feasibility of having laboratory closer, so developing mobile unit to get as close as possible to the investigation site. We also reduced the time to identify the causative agent, and the average calculation was minimum 3 weeks, but more between 3 to 6 weeks, which is a lot in outbreak. Also the algorithm approach, because when you have the platform, and the platform, you can, instead of working for individual disease, you always consider the syndromic approach like fever and rash, and you are testing for measles. If it's negative for measles, it is the same platform, just the reagent changing. You can test for rubella, you can test for whooping cough, you can add a gene expert and also test with the monkeypox. So having this cluster allowed us to identify rubella, but whooping cough, pertussis outbreak that are not often detected by reference laboratory. And it also improves the operational response. You are able to launch vaccination campaign at the right time and have the right impact. So to conclude, I think the mobile laboratory, we are convinced, and we try to convince everybody, that mobile laboratory are really key elements. You can have many laboratories for the response, for the investigation, and in the global health, but in global health crisis, I think the message should be getting the laboratory capacity as close as possible to the patient is really needed everywhere. It brings really the capacity especially in population affected in crisis situation, the modular approach and the capacity to switch from PCR to serology to genotyping allowed also quite a lot of flexibility. And we need to increase the numbers. I mean, if we have to say something, we should not fear, even in MSF, that sometimes we say, no, it's not our role. Yeah, we can play a role in this as well. And that's all. Thank you. Yeah. No question?
Hi, thank you very much. It's a general question maybe for the whole panel. Do you have a preference for point-of-care tests when you're considering, and also maybe consider, instead of testing the sample on site, maybe to save it in a matrix which is stable, and then you can transfer to a laboratory? Because if it takes 3 weeks still to organize everything to get there, then the timeline is quite precious.
I think the point of care, the problem is how long it takes to, I know in MSF we would like to solve all the problem of diagnostic by developing rapid diagnostic tests, but it's not possible and it takes so long. It's actually 5 years we are waiting for the measles rapid test, only these. So yeah, it can be, it's just a possibility. What we need is a clear, I think what is key is to be able in less than a week, when you start the investigation, being able to get the results, and then that will help to plan the proper response. So this time is the key one.
Just a comment also, I do agree with your frustration, but I'm also thinking where could be this push mechanism to really encourage companies to develop more point-of-care tests, because they are very valuable, but somehow it is missing that interest from the companies.
But I think we try through WHO as well to give more visibility, because also when they work on target product profile, for example, so we try to influence, because even the technology, they need to understand that we are running under specific climate condition, stability of the sample, stability of the reagent. Most of the reagent for PCR are 12-month shelf life, so it's also difficult in low middle income country for the supply system, and this is what we try to push. That's all.
Okay.
Thank
I
you.
did not want to be so...
No.
Thank you, Laurence. Okay, so I think our countdown is getting closer to 0. Then I just hand over to you, Kim, and Kim is going to show us now what they do with the mobile labs in the Arctic,
Well,
or in cold places in
Canada.
Canada.
I don't think this is going to hold that. So apparently we have 5 minutes left. I cannot talk that fast. So I do have time? Okay, so bear with me. Forward. All right. So, hello to you all. I'm really happy to be here and be part of this panel. It's a lot of interesting conversations about mobile labs. I am not so happy about the fact that I made a spelling mistake there. So let's just ignore that. Apologies, and I'll move forward. Fortunately, I am able to say what my institution name is without spelling error. So I'm from the Public Health Agency of Canada. And within the Public Health Agency of Canada, I am housed within the National Microbiology Laboratory. So in Canada, health is a federated system, and the provinces and territories are in charge of health delivery and health services for each of their jurisdictions. And we as a federal institution are there to provide regulation, oversight, surveillance, emergency response functions, and some esoteric or rare detection testing for the labs that can't do it themselves. So in this case, we're talking about high-risk pathogens like the Ebolas, biothreat organisms like anthrax, those sorts of things which cannot be done in the provincial systems because they just don't see enough of them and they don't have that capacity. Of course, we act as surge capacity as well, so it should come as no surprise that the laboratories or the mobile laboratories are housed within the National Microbiology Lab. Okay, so here is kind of a scalable example of the mobile labs that we have available in Canada. Obviously, we have to operate from -40 to +40. I mean, that seems crazy, but Canada does get that cold, and we do deploy to places that does get that warm. So when we are constructing our mobile labs, particularly the hard ones, the ones on trucks, we actually have to make sure that they are able to withstand -40 temperature. That's a lot of insulation, because they can be deployed anywhere in Canada. And of course, on the other spectrum, we also do international support, and we deploy for some of the viral hemorrhagic fevers. And I'll kind of cover a little bit of these two pillars that we have going on in Canada right now. So these two pillars are housed within one division within the National Microbiology Lab, and that's called the Health, Security, and Response Division. And within that division, there's 4 sections. The ones that do mobilizations are those in bold, special pathogens. They deal with level 4, the viral pathogens. And then there's the bioforensics, assay development, and diagnostics section. Bad acronym. And that's the section that I'm responsible for. And I can't take credit for coming up with that acronym either, but it's a good one. Okay, so pre-2001. So we all know what happened in 2001. And before that, we didn't have any mobile lab capacity. You know, if we had to go somewhere, we would grab a bunch of things from the lab, much like you were discussing. You just get things from the lab and you can take it somewhere else, right? We had very early infrastructure. We had one glove box. And then, of course, the events of 2001 occurred. And in Canada, we started getting calls. You know, can you come to us and do detection? Because pilots are not going to carry potential anthrax isolates from the provinces to the center of Canada. Pilots have the right of refusal. So what did we do? We threw everything in boxes, we chartered a plane, and we went out to the province that requested support, which is fine, but it identified a huge gap, right? So a lot more things started happening, right? These are the Amerithrax attacks. We are all well aware of that. But then these pictures in the middle here are actual things found in Canadian apartment buildings, for example. So there's real things like this happening. Suspicious packages, suspicious powders, kept going on and on. So our security environment realized that there really is a gap, and they started developing mobile labs that could be moved across Canada to deal with these sort of things. So prior to the Vancouver 2010 Olympics, which were hosted in Canada, in Vancouver, we had a grant-funded project to basically build what was called Science Town. Now, Science Town meant that all of the science institutions that supported the RCMP, which is our national police, for CBRNE response, so chemical, biological, radiological, nuclear response, built these mobile labs together so that they could be interconnected, share some things. They'd all have the same sort of operations. And so we built 2 mobile labs at this time, which from the top is a fully certified level 3 laboratory. It had all the things I think that Florian had mentioned as well. You know, it had a showering area, class 3 glove box. It had, or it has, it's not gone yet. It has environmental airflow, negative air pressure, all the things that you would require for a regulated level 3 laboratory. And that guy is big. Its primary intention is to be moved across Canada and then sit somewhere for a large-scale event. So if the provinces need surge support, it could be moved there and it could be sat there. If there's a large biosecurity event, like a G7 summit, a mass sporting event like the Olympics, it could get moved there, sit there, and we could work there for about 2 to 3 months, which we've done in the past. You might ask, hey, what are we testing in there? And what we're doing is we're testing for suspicious powders, if anything's found in those sites, because we're there to protect internationally protected people. We also set up a network of aerosol sampling around secure zones as well, and we do the testing for that on the regular. The second tier of deployment within our lab is called the Mobicon. So it's a shipping container on wheels. We've seen images of these before. It's very similar to the CL3, except it doesn't have a shower area in it. So it still has the glove box, it still has the negative air pressure, all those things, but it doesn't have a level 3. This guy, though, can be moved a lot faster. So we can mobilize this in a military aircraft in about 2 hours. So it can get moved a lot faster, plunked down in a spot and used, or it can augment the CL3 if we have multiple sites going on. And we've had that in the past where we have political events that may be spaced across different provinces or across different cities, so we have to have 2. And finally, but not least, very important to us, is this, we're gonna call it the lab in a hockey bag because we're from Canada. And I feel like that holds true here as well. But it's just basically a lab in a bag or containers like what's been presented before. And for us, the lower picture there, that does about 200 samples for us in the field. So we took that to the DRC to do some screening for viral hemorrhagic fevers. So you might ask, what are our deployment models? There's several. Those trucks, as I already alluded to, were very much developed for CBRNE response. And so domestic CBRNE response is one of their modalities. The team that runs it is called the Microbiological Emergency Response Team. And again, this is part of the RCMP-led National Response Team. So primarily the responsibility there is biological response and site security. A lot of our work is involved with legal investigations. Those 2 news articles there are people that have actually they do have jail time. We've given people jail time doing investigations to support the RCMP. So interesting cases, if you ever want to look one up. The lady there on my left was caught crossing the border. She had mailed some ricin letters to Trump when he was in his first presidency. And this one on the right was actually a federal scientist who was smuggling Brucella to China to develop tests overseas. Okay, so the other mode of deployment here is the international diagnostic search support. We've seen a lot of that already. Basically, a bunch of containers can be deployed out somewhere, and then we have a team that often goes into the field to support whatever is requested for assistance. And that's been going on since about 2003, and it primarily rests within our special pathogens group. Their acronym is SPIDER, so that's the Special Pathogens Diagnostic Response. Not sure what's better, SPIDER or BAD, so many acronyms. So an interesting case that kind of puts it together for us was a request, 2018, and this had to do with tuberculosis within our northern communities. So tuberculosis is still a problem in Canada with disproportionate numbers in our northern populations. And because of that, the government of Nunavut requested assistance to run a mobile clinic for tuberculosis screening in northern Canada. In particularly a little place called, I hope I don't butcher this, Kikajarjuak. We called it KIK for short. This was a community of about 600 people with the highest rates of TB in Canada at the time. And so we were requested to supply a mobile lab to support the clinic that was going on. So this was a case of bringing equitable care closer to the environment, right? Same sort of thing. If we had to wait for samples to come out of here, we're looking at days to get on planes, if the plane can get out because of snow or storms or what have you. And then when it gets out, then it has to go through the normal laboratory routing to get the appropriate detection, antimicrobial susceptibility testing, the whole jazz. So we brought that stuff to this site. Now, logistical problems you would expect, right? This is no different than deploying to some remote place in Africa, except we're not worried about cold chain here. We're actually worried about warm chain, if there is such a thing. Okay, it's really, really cold. So -38 was one of the ground temperatures when we were landing, when we were going up there. I feel like that's very cold. Please feel free to disagree. We are dealing with incredibly small planes. We're dealing with snow, irregular weather. The planes were only allowed to take, one bag, 50 pounds or less. So you've got to mobilize that stuff up into these places. You're landing in runways that are ice. You have to unload the planes yourself, basically, get to the site yourself. There's a lot of logistical concerns, food, housing considerations. I've never been deployed where I was told to watch out for polar bears. That was a new one. So look out for shifting snow. So that's an unusual consideration for us. And then a lot of it is the same as when you're going to other remote regions as well, right? Like food security and costs. So we had 50 people coming in to do this clinic. We have to house them and feed them. Food costs are already insane up in these areas. So you could bring food in, but remember, you're only allowed 50 pounds. So you have to make those decisions. Fortunately, we do have a lot of structure in our deployments. So we were able to kind of get all this going in a very short period of time to turn this around. I think our request to get there was 7 days. So we had to pack everything up and get going. But we do have a lot of stuff sitting in our warehouse ready to go. Yeah. And then, of course, the lab work itself, consideration is there. So we don't have our truck here. We didn't mobilize our container lab. So we worked in a local community center. And when you work in a local community center, you have to be cognizant of all those things that you would have in a real lab as well, right? So secondary air, for example, how do we isolate the organism? You can bring pop-up tents with you. You can bring containment devices for that. So we can achieve that. I mean, you can use a regular fridge for a lab fridge, right? You just have to be very cognizant in these situations that people are milling around everywhere. So you have to segregate things or label them very well, which I think we're all very aware of. Benefits. So there has actually just recently been a publication on the benefit of this sort of screening, and it has been shown to be beneficial. In this specific one, and they're still going on to the smaller communities. Out of the 600 people that live there, we did screen around 500 people, and we did detect 12 new cases. So there was several people that were identified for latent TB treatment. So there was benefits to this for sure. And then just a quick other cold deployment, because I feel like Florian very much wanted to see the cold deployments. This one was actually when we used our level 3 truck in the beginning of COVID. So February 2020, I believe it was, when we got the request to mobilize to a military base in Ontario for all the repatriated Canadians from China. So let's take it back in time. Canada chartered some planes, they brought back Canadians, they put them in hotels for 2 weeks and watched and waited, and we tested them if they had anything that even remotely looked like COVID. And so we sat there for about 2.5 months. It also was very cold. I have never seen a frozen porta potty before, but that was a first. We did use our truck for this. We actually blew out both generators at this time. And then we also had to mobilize into a second site for a second set of planes and ended up working in one of these tents. And so this again is where that warm chain matters, because PCR equipment does not like it when it's very cold. So considerations there. And then as COVID went on, we also procured some CL2 trucks that could be used for higher throughput COVID testing as well, like at border sites or so on and so forth. So when I say high throughput, I'm talking like 500 samples a day. That's about as high throughput as you could make one of these things in our hands. I would be remiss to say, or not mention that how important exercises are, but in the interest of time, I will just say the same that Florian had said earlier, I think for me it was Mina, that you know, there's a lot of advancements that you can do when you do exercises. There's a lot of learning, and being able to identify new equipment and new technologies is very important. I think somebody asked about nanopore sequencing and Illumina sequencing. We do both. One is clearly more repeatable, but we have these exercises to actually teach that or to learn that. And then to go forward, just capacity building, networking. I did say that we blew out both generators during our COVID deployment for our CL3 truck. So over the last 2.5 years, we've been doing the requirements and procurement of 2 new CL3 trucks. And it's been fun. So if anybody wants to talk about that, feel free to find me afterwards. And now we're going through the testing stage of those vehicles as well right now. And with that, I will end. Thank you.
Hello, thank you for this nice presentation. How many people or staff can one CL3 truck accommodate?
Lab staff?
Yeah.
Lab staff, we probably have about 5 people in there. We usually do teams of 4 or 5, depending on what we're doing.
Okay.
Well, I'll just sit down. Thank you.
Okay, I think, so I want to thank all of you and myself. And and then I can see already that I think we want to wrap up this session, and I want to thank you also for coming. And we are here for the next days, so feel free to just come to us and talk if you want, if you have more questions. Thank you.